In the previous video, we have talked about membrane potential and resting membrane potential. Now, in this today's videos, we'll be talking about action potential and its I basis. Types of changes in membrane potential. So there are basically three types of change in the membrane potential. The first one is depolarization, which refers to the decrease in potential, that is membrane is less negative. Repolarization, returning to resting potential after depolarization. So here in this picture, the membrane is depolarized to minus 60 millivolts and again repolarized back to minus 70 millivolts. While another change in membrane potential is hyperpolarization, which is the increase in membrane potential, that is membrane is more negative than that of the resting membrane potential. So action potential. Action potential is defined as the transient change in membrane potential after a threshold or suprathreshold stimulus is applied to an excitable membrane. Excitable membrane includes the membrane of neuron and the muscles. So it includes, so after the stimulus artifact there is a latent period. And now what are the different stages of action potential? So membrane depolarized to threshold level, and then also known as firing level, and then there's a rapid depolarization, there's a overshoot, and there's a repolarization, and then hyperpolarization. Action potential is also known as impulse or spike potential. Action potential, or this electrical signal, is critical to the functions of nervous system. Nervous system receive, process, initiate and transmit all the messages through action potential, and all the muscles including skeletal, cardiac and smooth muscles, they initiate the contraction. What is stimulus artifact? Different kinds of stimuli may be utilized for excitation. It can be electrical, mechanical, chemical, thermal, sound or light, energy. Latent period. Latent period is the time it takes the impulse to travel along the axon from the site of stimulation to the recording electrode. Actually, it's a time period from the stimulator to the recording electrode. So this is the latent period. And after latent period, the level of depolarization from resting state till it reaches the firing level or threshold level is known as local response, which is the initial depolarization. And after the firing level, there is a full phase of action potential. Now, what is the difference between this local response and action potential? So local response, also known as graded potential, this is generated at the site of stimulation. They serve as short distance signals and their strength decreases with distance and time. These are not propagated, while other side action potential, once generated, these potential are propagated over a long distance, and there is no decrement or change in size and shape of the action potential. Graded potential die out over a short distance. However, they can be summated, means when their magnitude is sufficient enough, they lead to the generation of action potential. So these are some of the examples of graded potential: generator-receptor potential in cutaneous sensory receptor, in-plate potential in skeletal muscles, postsynaptic potential in synapse, pacemaker potentials in heart, and slow wave potential in GI smooth muscle. Basis of action potential of a neuron. Resting membrane potential of a neuron is minus 70 millivolt. Then initial depolarization and the threshold for the neuronal excitation is minus 55 millivolts. So there's a rapid depolarization in which there is opening of voltage gated sodium channel, thereby increasing the permeability for the sodium ions, and sodium ions enters inside the neuron. While at plus 30 millivolts, this voltage gated sodium channel closes and there's opening of another channel known as voltage gated potassium channels, thereby increasing the permeability of the cell membrane to potassium ion, leading potassium efflux. This leads to the repolarization phase. While after hyperpolarization or simply hyperpolarization, there's the potential is more negative than that of the resting membrane potential, as voltage gated potassium channel is slow in nature, means it is slow to close, so more of the potassium is efflux. This picture shows sodium and potassium conductance during different phases of action potential. So G stands for the conductance, and increase in conductance means increase in permeability to that specific ions. So you can see during the depolarization phase there is a high conductance for the sodium ions, and during the repolarization phase of action potential there is a decrease in conductance for the sodium ions while increase in conductance for the potassium ions. And potassium ions conductance is for more period after times. This explains the nature of voltage gated potassium channel, which is very slow to open and slow to close. The voltage gated potassium channels are sensitive to same depolarization that opens voltage gated sodium channels, but they open more slowly. Okay, this is the summary. So there's a stimulus artifact, latent period, local response. So local response is the initial depolarization phase, which is slow. It begins from the baseline to the firing levels, and the ion channel that is involved for the local response are leaky sodium channel and only some of the voltage gated sodium channels. And after threshold there is a rising phase, rapid depolarization. What is overshoot? Overshoot is the reversal of the membrane potential from 0 to plus 35 millivolts. So at plus 35, voltage gated sodium channels closes and there is a full phase opening of the voltage gated potassium channel, rapid falling phase. After depolarizations, it is the slower repolarization phase that brings membrane potential to the baseline, while after hyperpolarization, also known as hyperpolarization, it is the membrane potential undershoots, means goes beyond the baseline. Baseline goes beyond the below the resting membrane potential. So these are the some ion channel blockers. Voltage gated sodium channel is blocked by tetrodotoxin that is released from Japanese fish, puffer fish. Local anesthesia, procaine and lidocaine, this also blocks the voltage gated sodium channel. And you can see in the diagram A and B there is a changes in the shape of the action potential. The peak level is decreased while the threshold is also increased for the excitation in both cases. While the side voltage gated potassium channel is blocked by tetraethyl ammonium, TEA, in which there is a change in shape of the action potential with the prolonged duration of the repolarization phase. It represents the voltage gated potassium channel is blocked by TEA. So this is the end of our discussion. Thank you.